Hello, I'm Hélène Langevin. I'm the director of the National Center for Complementary and Integrative Health at NIH. This talk was given on May 19th at the Society for Acupuncture Research meeting and is called Acupoints in Acupuncture Research, Past, Present, and Future. I will review the historical usage of acupoints in acupuncture research, controversies surrounding this usage, with consequential obstacles to optimizing the integration of acupuncture research into the overall research landscape, and potential solutions. Acupoints are like a 2,000-year-old riddle that we have not yet solved. I actually love the expression on this guy's face. It's like he's saying, no matter how they try to figure out what these points mean, they never will, because I just made this up. And that certainly is a view that has been expressed high and wide among acupuncture skeptics, that acupoints are just that, lines drawn on someone's skin 2,000 years ago that have been used empirically ever since, but that don't mean anything, other than making patients believe that something magical is about to happen, essentially a theatrical prop for an elaborate placebo treatment. But let's pretend that the gentleman we are looking at is not kidding, that he's trying to convey something important with this map. But we're not totally sure what this is, and we want to investigate. What we are provided are dots, acupoints, on a map, which is the surface of the body. This is a common problem in geology, for example, where people collect soil samples or probe for underground aquifers in varying intervals, and then try to reconstitute the whole map. Now imagine that we need to solve a riddle, and we're given a grid like this. And we can see a certain set of dots that look like this. And we're asked to imagine what the whole map may look like, assuming that we have never played a game of chess. A reasonable guess might be this. But now imagine that our initial sampling set of dots is slightly different. Like this. Well, we might guess this instead. And if we had this, we might end up like this, which is still not quite right, but it's starting to look more like a chessboard. We could keep refining our guesses by revealing more rows until you get the whole picture. This example illustrates that the location of our initial sample influences what conclusions we drew and what we guessed next. Now, here is another example of a map with a bunch of dots on it. It looks like this might be a map of a city, and that the dots may be lining up with streets. So a reasonable thing to do would be to look at a street map. And this looks pretty good, right? So maybe we might want to confirm this by asking somebody to take a picture at each of these dots. And we might find that 80% of them have street lights. So we're done, right? But then a clever person might say, wait a minute, let's try to overlay another kind of map on this, maybe a map with water. And we see that a lot of dots are on water. And we realize that this is a map of Amsterdam. And the correct answer is that a lot of the dots are actually bridges. So this illustrates how not only the location of the sample, but also the nature of the measurement that we make can make all the difference in our ability to solve the puzzle correctly. If you look a bit closer, you can also see that there are plenty of intersections that are not bridges and that presumably have traffic lights too, such that the traffic lights are a red herring. If we had systematically looked for traffic lights when there are no yellow dots, we could have seen that. But we jumped to conclusions too soon because the traffic light confirmed our hypothesis. And of course, this is an example of observation bias. And when it comes to acupoints, researchers have historically stepped into these pitfalls with both feet. In fact, acupoints have been the bullet and chain that acupuncture research has been dragging along since the 1970s. Here's an abstract from a paper from 2018 that I co-published with my colleague, Dr. Peter Wayne at the Harvard Medical School. And I'm going to read you the abstract from this paper. Despite having made substantial progress in academic rigor over the past decades, acupuncture research has been the focus of long-standing and persistent attacks by skeptics. One recurrent theme of critics is that the concept of acupoints has no scientific validity. Meanwhile, the subject of whether or not acupoints exist or not has been given too little attention within the acupuncture research community. Failure to use clear terminology and rigorously investigate the subject of acupoints has hindered the growing legitimacy of acupuncture as an evidence-based therapy. In this article, we proposed that a coordinated effort is needed to improve the use of terminology related to acupoints, combined with rigorous investigation of their specificity and possible biological basis. Little did I know that when we published this paper a year later, I would be at NIH. And one of the first events that NCCIH organized after I started was a conference jointly organized by NCCIH and the NCI, or National Cancer Institute, on translating the fundamental science of acupuncture into clinical practice. Of course, I had acupoints on my brain, so I made sure to use the workshop as an opportunity to ask participants their opinion about acupoints. Are acupoints a useful concept for research? What is their value versus hindrance? Are they impeding progress? Should they continue to be used in research? What transpired is that you can think of acupoints in two different kinds of ways that are not mutually exclusive. You can think of them as a set of distinct anatomical locations where needling produces therapeutically useful effects. And according to this definition, each acupoint can be described according to its unique anatomical address that determines its physiological properties. The second way to think about acupoints is a set of anatomical locations that are linked together by special shared attributes that differentiate them from non-acupoints. Because there was at the time, and still is, currently much more research evidence for the first definition, especially regarding the neural stimulation effects of acupuncture, we decided to focus on this at the meeting. What became clear was that the acupoint names are being used as anatomical shorthand to describe a specific location, and this was felt to be very useful. If you compare saying, on the anterior surface of the forehand, between the tendon of the palmaris longus and flexor digitorum longus, two anatomical units proximal to the wrist, compared with PC6, there is no contest as to which is the more convenient. So there were very useful conclusions that were drawn at the meeting. First, using acupoint terminology as anatomical shorthand is useful. It ties acupuncture research to traditional practice, and it facilitates communication between clinicians and researchers. Second, creating a standardized database to translate traditional acupoint nomenclature and conventional anatomy or physiology would be useful as a common reference for past and future research. Third, such a database can be thought of as a kind of Rosetta Stone, allowing classical knowledge about acupoint locations to be preserved and coexist with clinical and animal model data in a standardized way. Subsequently, NIH issued a request for information, or RFI, to gauge the effect of the community at large on these issues. More than 100 unique individuals and organizations, including SAR, contributed to the responses to the RFI and enthusiastically supported the idea of developing an NIH-sponsored open access repository and database for anatomical and physiological ontology of acupoints. Following this RFI, we are issued an RFA, or request for applications, for a research resource center to build an open access repository and database for anatomical and physiological correlates of acupoints using the U24 cooperative agreement mechanism. I was thrilled to recently announce that we will be funding the application entitled Topological Atlas and Repository for Acupoint Research, or TARA, with PI Dr. Vitaly Napadal and co-PIs Dr. Rick Harris and Dr. Carl Helmer. Very importantly, the Society for Acupuncture Research will be one of the performance sites, and Laura Triplett will be the program manager. This is an outstanding accomplishment for the team of investigators and for SAR. The goals of the project are, first, create an open interface for cross-referencing traditional and modern anatomical definitions of acupoint locations, and create essentially the Rosetta Stone. Number two, create a standardized body atlas with 3D coordinate system, which includes probabilistic localization of acupoints in relation to an origin reference point. Number three, create a database for multi-organ physiological data associated with stimulation at discrete acupoints and control locations. The project includes an administrative core and 4 interrelated functional cores. A traditional East Asian medicine or team core, led by Dr. Rick Harris, will collect and organize knowledge about the location and usage of acupoints from clinicians and acupuncture researchers, and will ensure broad outreach and implementation of TARA within the worldwide acupuncture research community. The mapping core, led by Dr. Peter Hunter at University of Auckland, will construct a 3D map of acupoints and their relationship to anatomical structures such as bones, muscles, and intermuscular connective tissue planes based on serially reconstructed MRI images. Dr. Hunter has long-standing expertise in standards-based physiological models and the use of computational methods for understanding and the integrated physiological functions of the body in terms of the structure and functions of tissues, cells, and proteins. The data core, led by Dr. Carl Helmer at Mass General Hospital, will build the acupoint database from the anatomical mapping data and integrate the anatomical data with existing physiological databases, such as stimulating peripheral activity to relieve conditions, or SPARC, the Human Biomolecular Atlas Program, or HubMap, and Open Neural. The ontology core, led by Dr. Marianne Martone at the University of California, San Diego, will build acupoint ontologies that define nomenclature systems, incorporating both the traditional East Asian medicine team and the modern biomedical anatomy system. I'm very pleased that the project will also leverage APNatomy, a sophisticated toolkit for visualizing and organizing multi-scale anatomical schematics and associated phenotypic information. I also want to particularly point out the importance of leveraging the knowledge developed in the SPARC program, which is an NIH Common Fund program aimed at anatomically and physiologically mapping the autonomic nervous system to assist in the design and testing of medical devices that can modulate peripheral nerves for therapeutic benefit. So the relevance of this project to acupuncture is obvious, with our increasing knowledge of connections between the internal organs and peripheral tissues. Indeed, one of the talks at the Society for Acupuncture Research, Dr. Kyu-Fu Ma, nicely illustrated examples of connections through somatosensory and autonomic reflexes, with different types of sensory neurons producing either pro-inflammatory or anti-inflammatory effects. And there is some additional very interesting and recent research that points in this direction. Here is a study of 2 animal models, stress-induced hypertension and inflammatory colitis in rats. In both cases, Evans blue was injected in the tail vein of the rat, and punctate areas of fluid extravasation and inflammatory infiltrates was observed in the forepaw for the hypertension model and in the hind paw for the colitis model. Electroacupuncture at these locations reduced the symptoms and compared with the sham intervention. Whether or not these locations correspond to acupoints or not, this study is important for several reasons. One is that it suggests a two-way viscerosomatic connection between the organs and the tissues along the corresponding dermatomes, which could explain one of the most fundamental aspects of acupuncture, that by stimulating points on the periphery can have effects on internal organs. And another noteworthy aspect of this study is that it points to the interstitium as an important area of study to understand the function of acupoints. That may tie this to the linked network system that traditional acupuncture texts talk about. And this is where going back to Amsterdam may be helpful. If we think of the nervous system as the rose, what kind of data might help us look into the nature of possible canals and bridges? In the case of Amsterdam, it was getting a picture of water. And as I hope to show you, humans are not too dissimilar. One type of experiment that has been around for a long time in studies of acupuncture has been the injections of various types of tracers into acupoints. But until recently, the result of these studies was difficult to interpret. However, I want to show you one study that, although preliminary, is nevertheless very interesting. In this study, a fluorescent dye, as opposed to a radioactive tracer, was injected intradermally at the PC6 point on the wrist and imaged sequentially. The tracer migrated towards the elbow at approximately 2 centimeters per minute. They did additional experiments using ultrasound to follow the course of the fluorescent tracer, marked using a metallic wire, and determined that the path followed by the intermuscular connective tissue between the flexor carpi radialis and the flexor digitorum superficialis muscles, as well as described in acupuncture textbooks. They also compared the course of the fluorescent tracer with isocyanide green, which binds albumin and enters lymphatics, and found some differences. Fluorescein sodium on the left seemed to accumulate at the elbow and stop there, while ICG did not accumulate and went beyond the elbow. Although these results are preliminary and need to be replicated, they suggest that fluorescein sodium spread along intermuscular interstitium while ICG spreads to lymphatics. You can see here the fluorescein signal on the left and the ICG signal on the right. These results make sense in relation to the new evidence that the interstitium functions as a fluid-filled sponge. Large amounts of interstitial hyaluronic acid bind water to varying degrees depending on pH, temperature, mechanical forces, and the amount of binding to hyaluronic acid determines the amount of water that is free to migrate through the tissue. Dr. Neil Thies and his group at NYU published a much publicized paper in 2018 on the interstitium. And although this is not truly a new organ, it was nevertheless a very important finding that the interstitium constitutes a mesh-like network with approximately 100 micron pore size. And this is exactly the same pattern that my lab had found back in 2002 using high-resolution ultrasound, when we found that acupuncture needles create a whirl through this tissue. Interstitial fluid drains into lymphatics, which concentrate albumin and pump the fluid back to the venous circulation via the thoracic duct. Acupuncture meridians correspond to interstitial collection channels along superficial tissue planes and drain into deeper lymphatics. Some acupuncture points, such as PC3, may correspond to locations where tissue pockets allow interstitial fluids to accumulate. And interstitial pockets could be natural places for neurogenic inflammatory infiltrates to accumulate, causing tenderness and reduced skin conductions. And finally, acupuncture may essentially ring the sponge and encourage interstitial flow and clear the channel. So putting all this together, we can think of 3 kinds of fluid flow from the tissue back to the heart: venous flow, lymphatic flow, and interstitial flow. And interstitial flow is the least well described, but it's interesting to think about what could influence it. It has no valves or pumps that we know of, so it likely depends on things like gravity, body position, muscle activity, massage, acupuncture, and inflammation. So we can think of different imbricated models for acupuncture that would all fit together. A neural model where acupuncture needling does specifically has its effects through the nervous system, as well as a different kind of model where there may be a function of the interstitium in mediating the effects of acupuncture. So hopefully, you can see that these two models are not necessarily mutually exclusive. Here, acupuncture point specificity may be determined by the anatomical location in relation to sensory nerves, while in the somatic model acupuncture point specificity may be determined by the location of the needle in relation to the interstitial channels. And finally, you can see that the two models may coexist, where acupuncture point specificity may be determined by both the viscerosomatic convergent innervation and the interstitial channel anatomy. So things to think about in the future. Well, first of all, an AcuPoint database will be a great opportunity to synthesize different types of data to arrive at a comprehensive understanding of acupoint and acupuncture. And Tara will lay the foundation for this work. And finally, I want to point out that the interstitium is a very challenging area to image, but important to understand how to bring this whole puzzle together, both the somatic aspects of acupuncture points, as well as the possible effects of mechanical forces, including the axial and shear strain that is produced by acupuncture needle manipulation. So hopefully, our 2,000-year-old wise guy is smiling at us now, because this whole thing was not a joke. And he sees that we are on the right track to crack the code and solve the riddle. Thank you very much.